Our next speaker is Michelle Halbur with Pepperwood Preserve, and she's going to talk about impacts of habitat creation on vernal plant species Sebastopol meadowfoam.
Good morning, everybody. I'm really excited to be here today to share some of my master's research that I did actually out of Purdue University in Indiana. To give you just kind of a general sense of the reason why I went into this project is as restoration practitioners, we often go into mitigation projects with the goal of either maintaining genetic diversity or improving upon it. However, we don't really know if we're having that effect. So this is really a retrospective study where I looked back at the impacts of habitat creation on Sebastopol meadowfoam. And so with that, we'll get going. So first I'm going to go over what is a vernal pool, just real briefly, and talk about vernal pool mitigation on the Santa Rosa Plain in particular. And then I'll jump into my study species, which was Sebastopol meadowfoam. I'll cover its abundance and distribution, as well as a genetic spatial structure that occurs not only range-wide, but comparing natural versus created pools. And my study was recently published, so if you have more questions or need more information, it is available. Real briefly, California vernal pools are these shallow depressions in a grassland matrix, and they're usually underlined by a hard clay, hard sand soil that does not allow water to percolate. So in the winter months when the water fills, or the precipitation occurs, those shallow depressions fill with water. You have a standing water table. In spring months, the water evaporates, and we get these narrow windows of flowering, and we get these nice concentric rings that are very typical in the Central Valley. And then in summer, you often don't see that there are vernal pools there, unless you do what you're looking for. And unfortunately, we've lost upwards of 95% of our vernal pool habitat, especially in the coastal regions. The Santa Rosa Plain is one coastal region where we do have vernal pool habitat. It's located in Sonoma County here. And this picture here is pretty much the entire Santa Rosa Plain. It's bordered by Santa Rosa and Contati to the east, Sebastopol to the west. And from this picture, you can really see there's a lot of urban encroachment, there's a lot of agriculture, there's modifications to the water system, so there's a lot of impacts that occur in this area. Mitigation on the Santa Rosa Plain for Sebastopol meadowfoam particularly started around 1999. This image to the left here includes one of the largest natural reserves on the Santa Rosa Plain. And you can see just to the east we have suburban development. There's major roadways to the north and to the south. And then to the left of this orange line, that's a recently created mitigation bank. So just directly across the fence line we have newly introduced seeds. Depending on who was the contractor and when they developed these sites, you have various levels of density. So here's a more historic or earlier created site where the upland to wetland ratio was pretty low. So a lot of wetland space versus upland space. And now it may not reflect what might have been more historical as far as distribution goes. When contractors go out and develop these sites, they need a source for seed. So they often go to natural and often created pools to collect inoculum by scraping the topsoils. They might mix multiple pools, take that inoculum across the range of the species and then distribute it among these pools here. To give you an idea of what natural pools and creative pools look like on the Santa Rosa Plain, here's a natural pool. You can see there's a lot of organic matter that has built up and we have invasive species moving in. However, you can still see Sebastopol meadowfoam, the white species here on the edge. Here's a recently created pool, about 3 years old at the time this picture was taken. And so with disturbance, it looks more typical, similar to your Central Valley pools with those nice concentric rings. But over time, it may eventually look like this without disturbance. So jumping into my study species, Sebastopol meadowfoam, it is an endangered annual herb. It occupies vernal pool habitat as well as wet meadows and swales. Swales are these linear features that water may move through. And for the purpose of this study, I lumped swales into the vernal pool classification. The species is primarily outcrossing, but there is evidence it could be selfing. It is pollinated mostly by generalists, but there are solitary bees with limited foraging range. There is potentially limited seed dispersal. However, the seed bank is viable for approximately 25 years or so. So there's lots of opportunity here, maybe for localized adaptation and restricted gene flow. I want to point out that the plants themselves can vary greatly depending on where they're growing. This plant here has upwards of maybe a dozen flowers. However, here at this site, you can't tell where one plant begins and another plant ends. So it's variable depending where you are. The geographic range of Sebastopol meadowfoam includes the Santa Rosa Plain, as I mentioned earlier. There are also a few satellite populations across the Mitigation mountain range here. We don't know why or how they got there. However, I did just study one of those. I sampled one of those sites as part of my study here. And if you're curious where Pepperwood is, we're right about here in the heart of the Mitigation. Okay, so I'm going to jump into its topographic distribution and abundance. My specific research questions were, what are the current patterns in 2009 when I conducted the study of cover and topographical distribution within pools? So where do they occur along that hydrologic gradient? And then do these patterns differ among individual pools or pool types, comparing created to natural? Here are my sampling locations. Created pools are indicated by the red triangles, and you will see that there's going to be a higher density of created pools that occur here. That's just where mitigation has taken place on the plain, so that's where we were able to sample. We sampled 30 to 50 individuals, and where limnanthes occurred is where we took our cover estimates. We also measured their relative elevations within pools relative to the edge of the pool, so the depth. And what we found is that on average, the mean percent cover of created pools was significantly lower than natural pools. When you look at this, though, on a pool-by-pool basis, we have created pools to the left of this hatch mark, natural pools to the right. You can see it's really due to a few features that are natural. So it's not a consistent pattern among the natural pools. It's just due to a few. Looking at depth, so 0 would be your pool edge. Well, we found that created pools, the limnanthes that occurred within those habitats, were found at significantly shallower levels than natural pools. And again, that's due to a few unique features. This here is a feature that actually had a lot of organic matter built up, and so it was convex versus concave. So in conclusion, the created pools we sampled had populations that were less dense and found at shallower positions. That shallow position could be due to the fact that it's occurring around the rim of the pool, or it could be due to the way that these pools are constructed. They might just be constructed like less deep. Specific natural pools and swales did provide high-quality habitat. Here's a swale here where the plant is actually, it looks like it was named after. It's foaming out of the meadow. However, none of the created pools we sampled exhibited similar quality to those natural features. Moving on to the genetics. I mentioned before that there's opportunity for naturally restricted gene flow in this species due to its fragmented populations, localized seed dispersal. These nutlets, it's speculated that they might float and distribute through water. However, they don't float very well, so it's most likely gravity dispersed. And then there's limited pollinator foraging range. However, through mitigation, we have this opposing force of artificially increased gene flow where through the design of high-density sites, we may be increasing gene flow among pools. We're moving seed across the species range. And this is, remember, the entire species range, with the exception of Napa County. And then there's the potential for disrupting any structure that may occur within pools if you're collecting seed from multiple seed sources, mixing it, and then spreading it throughout multiple pools. So my specific research questions were, at what spatial scale do patterns of just genetic structure emerge? Is it on the pool level, site level? What level does it emerge? Are there patterns of fine-scale genetic structure within individual pools? And then do these patterns differ among individual pools or pool types, comparing created to natural? And I want to mention I'm building my research off of work that was done by Deborah Ayers and Christina Sloop. And my sampling design just allowed me to ask that within pool level of structure, as well as to compare the mitigated sites to the natural sites. Here are some of the programs that I utilize. I want to introduce this concept of isolation by distance, which basically if you're an outcrosser, it means the individuals that are closer to you, you're probably more likely more related than you would expect versus individuals far away across the landscape. Here are my sampling sites for the range-wide study. Again, the most dense areas where there was mitigation is in the center of the range here. I sampled 29 pools across 21 sites, including Napa County, which is not shown here. I sampled a total of 614 individuals and analyzed 6 neutral nuclear microsatellites. And then where we took our samples, we measured its relative position within the pools as well as its elevation. Here's the structure output. We found that there are most likely 4 genetic populations. Right away, you can see Napa County. The most isolated population we sampled was, it clustered very strongly by itself. We have 1 isolated peripheral population on the Santa Rosa Plain. 2 of the youngest created pools actually jumped out, and it actually clustered with a few other natural features. And this is what it looks like on the Santa Rosa Plain. So Napa County is not shown. Here's that isolated population that is its own genetic cluster. And we have another cluster up here and then the remainder clustered together. What's interesting is this northern cluster, it was really driven by these 2 created sites. Those are the 2 youngest created sites that were actually in that first picture I showed you of the largest natural reserve here and that newly created site on the west there. The seed source for that is from a northern site that I did not sample as part of this study. When I spoke to the contractors, they mentioned that they sampled those northern sites for a different species. They weren't sampling for Sebastopol meadowfoam. However, there is Sebastopol meadowfoam present, so they could have brought new alleles closer towards the center of the species range, which caused this cluster to pop out. Or it could be due to the fact that at that location, there might have been seed bank already present, and through the mitigation activities, they might have agitated the seed bank and caused germination. But when you remove these 2 from the analysis, this genetic population disappears. Looking at isolation by distance across the species range, across your x-axis here is your distance class, so how far away are you from other individuals. The red hatch marks are your 95% confidence intervals, and any time the blue mark is outside of that is a significant finding. And what you see is that individuals from 1 to about 3 kilometers are more related than you would expect by chance. And the further away you get, the less related you are, with Napa County being the most isolated. This just means, this flat line is that there are no pairwise comparisons at those distance classes. When you look at the Santa Rosa Plain alone, you comparing natural pools to created pools, you see that same trend in natural pools. However, in created pools, at 3 and 4 kilometers, you see that they're more significantly related than expected by chance. And so most likely we're seeing contemporary levels of elevated gene flow due to those activities. It could also be due to collecting seed from the same source populations and spreading that throughout the center of the range. Moving into the within pool structure, here are my sampling sites, including Napa County. We conducted the same isolation by distance analysis within pools, and we didn't really find any patterns. Some pools exhibited it, some pools didn't, and it didn't really matter if it was a natural or created feature. And then we were also interested in asking the question of, is there structure along this hydrologic gradient, so that micro-elevational depth? And we did that by conducting partial mantel tests. Again, what we see, these 2 natural pools were significantly, they had structure, so depending where you are in that gradient, it was correlated with your genetics, as well as this one created pool. But there was, again, no consistent trends between created or natural pools, indicating that individual pools are very unique. So in conclusion, spatial structure emerges at a larger scale than most mitigation or conservation delineations. And what I mean by this is it's not just a pool, it's not just a site with a fence line, it's beyond that, and that it appears there are 4 genetically and geographically significant populations. We did detect significant range-wide isolation by distance, meaning that there are natural barriers to gene flow that are occurring. And so when we're moving seed around, we really do need to take that into consideration. Created pools do have evidence of elevated levels of gene flow. This could be due again to the density of pools, to seed movement and mixture. And there, with these pools being so different and unique, there is the opportunity potentially for local adaptation to be taking place. So by moving seed, concentrating it in the center of the species range, we may be artificially homogenizing the genetic variation. And then the patterns of fine-scale structure within pools, it was different among the pools. So my final conclusion is that individual pools are unique, like snowflakes. So some recommendations we had, in particular with monitoring, was that it was really inconsistent with what we saw on the ground. And I want to mention that there is a habitat conservation plan out right now, a draft for the Santa Rosa Plain. It includes 3 endangered species in the California tiger salamander. And U.S. Fish and Wildlife pulled a bunch of scientists in and consultants in to talk to us. So we did provide these comments up front. However, I still need to review it to make sure they incorporated it. But really, currently, an example of monitoring might be looking at percent cover, plant counts in a 5-year window. They really need to be looking at plant fitness and reproductive outputs and incorporate a longer monitoring window. And then implications for mitigation, we suggest that because the natural features have more heterogeneity, it might be useful for this particular species to incorporate swale habitat or other diverse, wet meadow habitat in mitigation plans. We do need to consider the biology of the seed sources. We need to protect those peripheral populations because they are a source of genetic diversity. Documenting seed movement, monitoring those source pools. So how are we impacting pools that we're regularly collecting inoculum from? And then monitoring pools adjacent to newly created pools. So those 2 at the northern site, the 2 created pools, it's right next door to the largest reserve. How are those unique alleles or genotypes going to affect the neighboring pools? Because we know that gene flow happens at a larger distance than the neighbors. So with that, thank you very much.